Thank you very much for inviting me to this conference. You will excuse me if I skip the first slides a bit too fast, mainly because Maria touched upon some issues anyway so elegantly, much better than I could do, and because we are running out of time, so I will try to make it a bit fast. So aniridia is the name that we have for this condition, which is essentially is a misnomer, even though it is the wrong name. It comes from the Greek words meaning absence of iris. And depending on the severity of aniridia, you can have phenotypes on the eye ranging from having an iris that doesn't seem any different at all up to a completely lack of iris presence. This is a structure of the eye in a more simplified form. So essentially you have the cornea on the top and the iris, that is the colored round part of that. And through the lens you have the light that comes into the eye and it hits the fovea, the bottom, the lower part of your eye. And next to it you have the optical nerve, which is responsible for taking those signals from the cells and transporting them to your brain and making those images. PAC6 is the only gene that we know that mutations on that genes are responsible for the aniridic phenotype. However, it is very well conserved on a genetic level, and what that means is that you have an almost exact copy of that gene in other animals, and that's helpful to us because essentially it allows us to use those animals as a study model. And the animals, the organs that are mostly used, essentially is the mouse and Drosophila, which is a fruit fly essentially, and the little fish called zebrafish. Aniridia comes along with other phenotypes as well, so it's not just the lack of the iris that's present. It affects the whole of the eye, and those features can manifest themselves in different time parts of humans. It can appear from birth and infancy up to childhood through the middle teens and adulthood. The major ocular features of aniridia is corneal diseases, glaucoma, cataract, dislocated lenses, defects on the iris, the foveal hypoplasia, and optic nerve hypoplasia. And also patients with aniridia have a poor vision and nystagmus. Cornea essentially is the part of the eye that is responsible for refracting the light source, and the physiological cell of cornea is very important for having a good vision. It's composed of three different cell types. The superficial, cell type, yes, and you have the first layer, which is the superficial layer, and then you go with the wing cells that comprise the third layer, and you have the basal cells and the basal membrane. I will skip that. The corneal epithelial cells originate from the stem cells, which are located in the limbal epithelium, and essentially the misbehaving of those cells is one of the major sources of the problems we have with aniridia. However, there is a mouse model that we are able to study with, developed by Dr. John West and Professor Collinson back in the 80s. So from a mouse, so you have a PAC6 mutated mouse that shows similar phenotypes as a human with aniridia. And one of the main issues is the keratopathy that is related with aniridia, and the therapies that we have for them so far are those in this list. So we can try anything with tinted or photochromic glasses, up to the use of lubricants, either preservative-free or with hyaluronic acid. And then you can use eye drops, serum eye drops, or different amniotic membranes. You can either have some limbastel stem reconstruction, either from living donors or non-matched organ culture donors. And you can reach up to the point of corneal surgery. None of those is the best therapy that you can actually have so far, at least we think. So what we are trying to do in the labs of Liverpool University is to develop a therapeutic tool for aniridia and associated phenotypes using morpholinos. And what morpholino is will become evident in a bit. First, I would like to talk you through the molecular dogma, and this is how scientists explain the flow of information into our bodies from cell to cell. So essentially, you've all seen the typical structure that we have in our minds about our DNA, a double helix that consists of 4 different letters, 4 bases, and that combination, that sequence is what makes our genetic material. Now, depending on the needs of the cell in different points, parts of the DNA unfold. Essentially, that parts are what we call genes, and they, through a process called transcription, they copy the cells to another molecule called the messenger RNA. The RNA is pretty much similar as the DNA. It still has 4 letters as a sequence. The major difference is only one chain. That RNA is what carries the message from the DNA up to the creation of the proteins, because every 3 letters of the RNA molecule are responsible for encoding one amino acid. The next 3 letters encode another amino acid, the next 3 another amino acid. And that chain of amino acids is what we call a protein. And this is where things can actually go wrong, because if you substitute one of those letters with a different one, you have a mutation. Now, depending on that substitution, a mutation might have no impact at all. It might produce a protein that doesn't work as well as the wild-type protein, or might not produce any protein at all. Humans are diploid organisms, so essentially they have 2 copies for each gene, each from one of your parents. So what we call a wild-type homozygous state is when both of your copies produce your protein, and that's the homozygous wild type. If you have a mutation in only one single copy of your gene, then you are in a heterozygous phenotype. The other copy is producing the normal protein. And if you have a mutation in both copies, then you have a homozygous mutatic phenotype where there is a chance that you don't have any protein made at all. Aniridia falls in the middle category, where you have a mutation in only one single copy of your gene, the other copy works as intended. And if you are a heterozygote, if you have a mutation in one of your single copies, it means that you don't make enough protein. If that loss of protein concentration leads to an abnormal or diseased state, it's called haploinsufficiency, which essentially means not enough. And aniridia is haploinsufficiency. Haploinsufficiency in general can lead to developmental disorders, metabolic disorders. They are responsible for some type of cancer and epilepsy. A set of genes that is strongly correlated with haploinsufficiency are called transcription factors. And what they do, essentially, they regulate the rate of transcription, how fast and how much you make of proteins of other genes. And because of that, they are found mainly involved during the development of organisms. And because of it's due to that nature that they are strongly correlated with haploinsufficiency, because it will mean that even small changes on your total protein levels can lead to a cascade of events, leading in the end to an abnormal phenotype. One of those transcription factors is PAX6, which is the only gene responsible for aniridia. It's a master control gene for the development of the eyes, the pancreas, and the central nervous system. And it regulates different cellular processes, including proliferation and differentiation and cell adhesion. As a transcription factor, it has other genes that it targets and regulates, including itself as well. And most of the mutations that we know of PAX6 lead to either a loss of function of the protein, or no protein being made at all. The only good thing, as I said, about PAX6 essentially is that it's highly conserved, so you can use other animals to study its effects, and that gives us a lot of opportunities, to be honest. There are over 800 mutations so far being reported, considering PAX6. Less than 500 of them, about 500 of them are reported only once. That means that we don't have yet a clear understanding of which way the mutations can lead to an aniridic phenotype or not. We need more data. So part of our project is actually trying to gather all that data, put them together, and see if we can actually visualize some patterns emerging, depending on whether those mutations are on a specific part of your protein, or is it the specific mutations that seem to appear more often. And we don't know exactly the technicalities of this, because there have been reports that even of the same exact mutation can lead to a different phenotype. So we know there are other things involved as well. The good thing with our therapeutic tool that we're trying to develop is that we don't really care about the mutation. Because we only target the wild type copy of your gene, the one that gives rise to the proper PAX6 protein. So it would make sense for someone to think, okay, if less than nominal levels of PAX6 are the cause of aniridia, so if we were able to increase those levels, it means that you will be able to slow the progression of the disease, or even stop it. And how would you be able to actually do that? There are different ways that you can increase the expression of a gene, at least on the cellular level. It's good to know, though, that cells make a lot of RNA, and they destroy a lot of RNA as well. And much of that RNA, they don't even use it sometimes. So it's a natural, continuous process that happens. So if you were able to go into that process and stop or lower the rate that the RNA is destroyed, it would mean that slowly, with time, you will have more RNA accumulated into your cells, and that would give more time to the mechanisms of the cells to make that RNA into a protein. Again, different pathways of how mRNA is decayed into the cells. One of those involve what we call microRNAs. They are small RNA molecules, only 22 bases long, 22 letters, and they have the ability to target a specific point of the mRNA and bind onto it. That binding attracts more proteins that they form a complex, and that complex can either destroy your RNA, that specific RNA, or without having any effect on the RNA, can block the translation of the RNA to proteins. The final effect is the same. You have less proteins than you need to. So it makes sense that if you are able to take that microRNA out of the equation, you have more RNA available into your cells. You can target directly the microRNA, but that is not easily advisable, because every microRNA has tens to hundreds of genes as targets. So essentially, you will be messing up with the cell. What you can do actually is protect the site of the RNA from microRNA binding onto that. And that's what we are trying to achieve. So for example, the lower sequence is a small part of your RNA. The top one is the microRNA. It only needs to recognize those 6 letters in able to bind onto the RNA. That's it. So what do? We take the sequence of the RNA and we scan from left to right of the seed region, that's how it's called, and to find which sequence give us the best results. Which sequence will give rise to the best oligonucleotide, the best sequence that will bind more strongly than microRNA. And then we send it to a company, and they synthesize that molecule for us. And by putting that molecule into the cells, it binds on your RNA in that region. It doesn't have any effect on PAX6, but it disallows your microRNA to bind onto there. So you have slower degradation rate of your RNA. And for that, we use morpholinos. That's what we call morpholinos. Small molecules, synthesized molecules as well, 25 bases long, maybe, depends. They are like DNA, essentially. You have the bases, 4 bases in the sequence that you tell them to. The major difference is that the chemical backbone that keeps those bases together is synthesized in the lab. So it's not recognized by your cells. So it's not degraded. It's not the target of your nucleases, and doesn't invoke any immune responses to the cells. And another positive thing of morpholinos is that they are easy to deliver inside of the cells. So essentially, as you have your cells in a colony, in a medium, you just put the morpholino into the medium, and the cells passively take that medium into them. Picking the right target, picking the correct microRNA to block, though, is not as easy. Because there are online databases that tell you which microRNAs might have a specific gene as a target. But if you go and look at that, you might come something like that. It's a small part of your RNA, and those little numbers are the different microRNAs that might bind onto your mRNA. Obviously, you cannot work with that. So what we do, we go to the databases, and we check. It's coming a bit funny here on the slide. Anyway, so essentially you check which microRNAs are believed to bind onto your RNA, and you come up with something like 2,000 different microRNAs just for PAC6. And then you're trying to eliminate microRNAs with a cleanup process. So you're checking the scores. How well do they bind on your RNA? How well conserved they are on other animals? Because you need other animals to study the effects. And then you check whether is there any experimental evidence of those microRNAs binding onto PAC6. And then with different criteria, we ended up choosing 3 microRNAs, depending on how many sites they bind on PAC6 and how well they bind, or how much they are expressed into the cells. We also check the literature to find microRNAs that already know that they have an effect on PAC6. And then we started going down and see how well they bind, whether they express on the tissue that we care about, and whether they are conserved as well. And we picked 3 more microRNAs. So we had 6 microRNAs in total to check. And this is where the results section comes in. So essentially what you do, you have your cells in flasks. You always keep a flask with cells that you don't do anything to them. These are the untreated cells. And the other flask, you put different concentrations of morpholinos in different time points. And when you want to, you take the cells out, you open them up, extract the PAC6 and see whether they have more PAC6 than the untreated cells, less or no change at all. One of the first experiments we tried in the lab with PAC6 was to find out how fast it degrades into a cell. So essentially there are chemicals that we can treat the cells with and stop the transcription process. And by measuring by that point, what we call time point 0, that you start the transcription, the levels of PAC6, you can see how fast it decays. So by measuring PAC6 after just 1 hour, for some reason, instead of going down, it went up. Probably that should be an indication that it's not going to play ball. Anyway, but however, if you kept the treatment going on, you could see that with time it started decaying, it started going on, it started disappearing from the cells. It took 4 hours to reach the wild type levels, but after that, every 2 hours it started going down like 50% every 2 hours. And when we treated the cells with morpholinos, this is what we start getting. These are 3 graphs, each one a treatment of a different morpholino. From left to right is the concentration, so the third bar of each graph is the highest concentration. Everything that is above the line, it means that you have an overexpression, you have more PAC6 than the untreated cells, and everything that is under the line means that you have less. So what we noticed is that although there was a tendency, a trend, for the PAC6 levels to be elevated, because it didn't seem to be dose dependent, because you expect the more blocker you put in, the more PAC6 to get, and because the error bars, those little things on the bars, were quite large, is what we call a non-significant results, something that we cannot trust. So maybe there is a trend with those, but nothing for us that we can actually work with. Then we moved on, and some better results came on. So we see a clear dependent response of the cells with these 2 different morpholinos. And even though the change might look small. I mean, in the first graph, essentially, is like 20-30% tops increase of your PAX6. Remember, because we are talking about a transcription factor, even 20% increase might be sufficient, might be important. There are clinical trials of drugs that have proven on their genes that they only cause 20% increase in expression, and that's enough. So we don't looking for something massive, we just need something that is consistent. And our best target so far is with miR-36, which on the highest concentration, it showed a twofold increase of PAX6. To put it into easier terms to understand, is that if those cells were from an aniridic cells, from an aniridia patient, for example, they were able to reach the wild type levels of PAX6, at least on the gene level. One thing that we scientists tend to do is trying to disprove ourselves. Every time we are not able to disprove ourselves, it means that our results are strong. And elevated levels of PAX6 are good, but you want to make sure that what you see is real. So one way of doing that is checking the targets of PAX6. We said that PAX6 is responsible for controlling other genes. And since we saw elevated levels of PAX6, you expect to see the other targets change accordingly. One of those targets is keratin 10, and you know that whenever PAX6 goes up, you expect keratin 10 go down. And we checked on the cells, and that's what we find out. So with the same concentrations, we have like an over 90% decrease of K10. You know, that's a really positive result. It means that what we see is real. More interesting, though, is the fact that when we checked the treated cell with the other morpholinos, the morpholinos that seem not to work as well, keratin 10 levels did go down as well. In some cases, even more. We reached a point that we weren't able to detect those levels. So that gives us an indication that you might actually do a difference into your cells, but it's not observable on the RNA level. You have to check the proteins as well, because in the end, that's what's going to affect the cell, the phenotype of your cells, and that's our next step. So by picking the best target so far, we also checked how the protein of PAX6 changes. And this is the way we visualize PAX6 levels on a protein level. So we just treat the cells the way we want to. We open them up, we extract the proteins, and we run them onto a gel, and it separates the proteins. And we know that in this case, this black line is the PAX6. The more intense the signal, it means the more protein you have. So the left column is the protein made from untreated cells. And the next three columns are untreated cells with morpholino or higher concentrations. And we saw that the signal gets more intense and more intense. It means you have more protein. And we need to put that into numbers. So obviously, we don't trust our eyes for that. We use software, and you get something like that. You get 2 times more protein than the cell was actually able to make on itself. And these are data that we need to actually, these are positive data that will help us move on to the next steps. And this is, by having those data, what we need to do now is gather all the information we have for the morpholinos that we already have, check the impact they have on the proteins, and then what we want to do is redesign them as vivo morpholinos, as they called. So essentially, it will be the chemistry that will use them for a clinical trial. And by picking those best targets, we will check again the protein level difference that they have. We will check other targets of PAX6 to see if you get the changes you expect to. Obviously, you have to prove that whatever change you get is due to the PAX6 levels and not something else. So we're trying to prove that there are no side effects as well. And we have a cell line that is PAX6 haploinsufficient. It has a mutation on one of its copies, so it doesn't produce enough PAX6. We're on 1 minute. We're going to use the morpholinos onto that cell line. And if the results are in the same level as the ones that we had so far, and they are positive, we will move to a mouse model. So essentially, we'll get mice that have an aniridic phenotype, and we will try to treat them with the morpholinos and see what difference it would make and whether we are able to stop the progression of the disease or slow the rate, or who knows. And also that's a good point where you're trying to figure out how you're going to deliver your drug into the eye, because that's a different hurdle as well on its own. And that's where we are now. This is the team working in the Liverpool University. Dr. Kevin Hamill, Professor Colin Willoughby, and Dr. Sajid Ahmad. And thanks to Dr. Daniel Aberdan for providing us the cell line with PAC6 haploinsufficiency. This research so far is funded by Fight for Sight. And I would like to thank you for inviting us to talk here. Because we as scientists, we tend to lose ourselves into the labs and having our faces stuck in front of a PC screen. It's nice to be reminded of why we do what we do and for whom. And, congratulations to every one of you for organizing this event and for the community as well. So thank you very much. And if you have any questions, please feel free.
Thank you very much to Thanos and to everyone at Liverpool. Fantastic.